Today, we are discussing with Dr. Rose Ruiz Daniel her recent PLOS ONE Lab Protocol article entitled A Versatile Nuclei Extraction Protocol for Single Nucleus Sequencing in Non-Model Species, Optimization in Various Atlantic Salmon Tissues, and the associated protocol in protocols.io. Dr. Ruiz Daniels is a research fellow at the Roslin Institute in the University of Edinburgh, working on elucidating the functional basis of sea lice resistance in salmonids. Before, she held a position in integrative fish genomics, also at the Roslin Institute. She obtained her PhD on population genetics of conifers in the Forest Research Center in Madrid, Spain, and her master's in applied ecology at the University of Manchester. Rose, thank you so much for being with us.
Hi, thank you for inviting me.
So to kick off, can you briefly explain what this protocol is about, how to use it, and why it is relevant?
This protocol is to extract nuclei from frozen tissue for the purpose of single-cell sequencing. And the main the main tissue that we use this protocol for is fin and skin and Atlantic salmon, but it can be used for most tricky cell tissue types. And apart from brain and sole female gonads, those are the only two tissues that we've tried so far that it hasn't worked. The main step is to be able to get something that has been flash frozen, and that's really important, tissue that has been flash frozen, and take it from a flash frozen tissue that is stored in a -80 into a single cell RNA-seq prep without having to use fresh tissue. And it's optimized to work in tissues that are going to yield very poor quality samples using other nuclear isolation methods.
And just to understand the relevance, can you elaborate on the state of the art of RNA extraction from nuclei when you decided to optimize this protocol? So why was the optimization needed for the field?
Okay, so when I started my second postdoc in Roslin, the motivation was we have this really big sea lice challenge with 4 different species of salmonid, some that were resistant to sea lice and some that are susceptible to sea lice, including Atlantic salmon, which is the biggest farm species in aquaculture in Scotland, in Norway as well. So it's really important in Scotland, and sea lice is a huge problem that they have. So we wanted to see at the cellular level why some species of salmon were susceptible to sea lice and others weren't. But we had a huge problem here. Sea lice infect the skin, and they infect the fins. A disease-challenging experiment that requires multiple species that had to be done in Prince Edward Island in Canada is you can't use fresh tissue, you can't use cells. But then also we had done practice sample runs with fins and skin of Atlantic salmon in Roslin here in Edinburgh, and we'd found that these practice libraries were not coming out great. We were losing a lot of sequencing, and sequencing is very expensive, and a lot of quality. And when we want to look at the cell types involved in Atlantic salmon resistance and compare it to that to the susceptible, you need really good quality data. So how do you get good quality nuclei out of these very tricky tissues? In my previous postdoc, I had to optimize an ATAC-seq protocol on frozen tissues for aquafang, and we had tried lots of different nuclear isolation techniques. So I got my favorite one from that and from previous work that I had done in Atlantic salmon, and just tweaked different things to see how I could get better data quality from these tissues, and that is what motivated me to do this protocol. So this sea lice experiment was completed a year ago with I think we did nearly 100 single cell libraries on these 4 species using this protocol. So that was the main motivation for it.
And I was curious, you mentioned that you have tried many tissues, but it hasn't worked in brain and female gonads. Is that correct?
Yeah, female cell gonads. Yeah.
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 Any wild guess as why that could be the case and how potential user could play around to get it work in those tissues?
So with brain, it's because it's got a lot of fat in there, and I think that's a problem when you're trying. You've got to use a different method, more like it's still with female gonads in the soul. I'm not sure. I reckon you could do about a week or 2 optimization, but I think the things that are being used for brain might work for female gonads as well. It just required more time because I was focusing on the sea louse and the fin, and for the fin and skin, I probably didn't have the amount of time, but other people have then used this protocol and compared it to other ones, and they found that there are things that work for those tissues.
And for those tissues that work, can you let us know what in the protocol are the most critical steps?
And this is a good one, because the most critical step of all of that is sample preparation and flash freezing. If you prepare the sample and the tissue has been dead for a while, and before you flash freeze it, you can do a nuclear isolation and you can see immediately that the nuclei are not the correct quality. Sample preparation, amount of sample that you put into a tube, because it's much easier to cut out the tissue that you want as quickly as possible, flash freeze it, and then you have a minimal processing step, because you sometimes have to dissect on dry ice to get the amount. So flash freezing as quickly as possible is the most critical, followed by amount of sample that goes in the flash froze and that goes into the protocol. If you can optimize that, then you get beautiful nuclei. And the final thing that is really, really critical is to keep everything at 4 degrees. As soon as everything goes above, while you're working it, everything has to be cold and quick as possible.
And I noticed that at the end of your manuscript, you mentioned that even though you were interested in lice, in sea lice and the interaction with salmonids, you mentioned that this protocol has been successfully used in many different species.
Yeah.
Including chicken and mice and sharks and rabbits and soles and pigs. Has there been any further optimization or tweaking of the protocol to get it work on those species, or is it exactly the same protocol?
So every new species and tissue, I would recommend running the protocol without the RNase inhibitor, because there's like, for example, and this is in the protocol itself, what the filtering steps, what size filter do you use, and then also what is your what sequencing what library construction platform you're going to use. So for Tenex, which is what we use for the sea lice, it's quite important, the filtering step. But for example, if you're going to use another library preparation kit, like we've now optimized this protocol for PASS, then you further optimize that. You can optimize it differently, because the filtering step is not as important, because it won't mess up with the microfluidics. So this protocol is currently being used in the Genomics Center at the Roslin. So they're constantly optimizing and changing things. What I use it for, aside from the sea lice, is my other work, which is on wound healing. So I used it for a wound healing time course in Atlantic salmon, which I'm currently working on. And one of the things, the final buffer, which is PBS plus BSA, an RNase inhibitor, the concentration of that, that can vary per sample. And that's important, because the amount of cells that you get at the end is critical for when putting it in the different sequencing platform, or not in the different library prep methods. So I would really pay close attention to that. if you're optimizing it in a different species. As for species, we currently have three manuscripts under review. Two have just been accepted. Two of them on Atlantic salmon, and one is the sea louse manuscript. And these are all using this protocol. And then I'm currently working on I'm just finishing characterizing mesenchymal stromal cells in the skin of Atlantic salmon, also using this protocol to characterize those. And in terms of like everything else that we've done, well, rabbit noses, sole gonads, sole noses as well. We've done sea lice and shrimp. We're using this. The shrimp and the sea lice require a lot of optimization. And once again, sample prep, I cannot, like, the amount of times sample preparation and optimization of that step is the best thing you can do for this protocol, but in general for any experiment.
Yeah, it seems that you have invested a lot of time on developing and optimizing this protocol for your original needs, as well as, you know, to extend it to other tissues coming from different species. Mm-hmm. So what was the motivation after all the work you put to go like one step further and invest time in getting the protocol peer-reviewed and published as a standalone methodological paper?
Well, we had a lot of interest in we did a review paper on how to design single cell experiments for aquaculture that then fed into non-model species. And we had a lot of people asking about this protocol, like how we done it. And one of the things that we found was sample prep was like one of the key issues. So having spent a lot of my postdoc in a lab optimizing this, I then thought, well, why would people just keep doing this? Like, why not put everything that I've learned into a protocol paper, and then I can just send it to people? And then and also we got asked for it a lot, so I thought, well, this is a good way. And also, as a early career researcher, you need to publish, and when you're in the lab, it takes a while to go from optimizing a protocol to a final manuscript. So I think this intermediate step really helps. Well, it's not really intermediate step. I felt that this was, I wish I'd had it. That's why I published it.
So yeah, I know what you mean. That is not an intermediate. This is a contribution, and it's a significant contribution, right? It's just the fact that it's based on a method and not on a biological question.
Yeah, but it was a biological question that motivated the protocol. Yeah.
But you need to optimize and to have the right method.
Yeah.
And can you tell us about the feedback that you received during peer review and how that shaped or not the final version of the protocol and manuscript?
So before sending this protocol to peer review, I would hand it out to people in the lab and ask them, how clear is this? And what is missing? And it was surprising. So what I felt that peer review was, what it helped is because, like, we're sort of in this salmon single cell world, and that kind of extended it a little bit outside there to other scientists with a different background. And what it really helped is to make the protocol clearer to a wider audience. So I found that really, and I find this for protocol papers, this is really important, because as soon as people see that something isn't accessible or easy, they get like scared of it, and they're like, oh, I don't want to do that. But this the peer review helped made that the protocol clearer, and also make how the results clearer. So that I found was really helpful. That's why I quite like the idea of it. Yeah.
And why did you decide to make the peer review history public?
I was like in two minds about that, because I always feel like, I thought in this case it would be good, because a lot of people have asked me similar questions to the reviewers as well, or will in the future. But I'm in two minds about that one. It was the first time I'd encountered this as well during my career, and I thought it would be good. But I can also worry that it would put people off peer reviewing, because sometimes you just want to have a conversation between the author and the between the author and the reviewers, and I can understand why, but it's also a good way for other scientists to learn how the peer review process works. So yeah.
And I think, you know, you mentioned earlier that as an early career researcher, it is important to publish. And I know this is a relatively young paper, although you've been working on the project, you know, for quite a bit of time. But can you tell us, or at least speculate, what do you think is the significance of having this peer-reviewed manuscript already published for your career?
I mean, for me, it has allowed me to be able to show that, I mean, because we're doing huge long-term experiments here, you know, things that involve cross-countries collaborations and experiments that take time, not just to optimize in the lab, but then computationally. So the thing, this is like a stop point between, well, you're in the middle of a big experiment, to be able to find out, show that you've produced something valuable while still doing these long-term experiments that can take years to finalize. So it gives you this, and then not only that, it also allows you to sort of get people to give you feedback on that part of your work. So when you do a next experiment, you can improve on that, and people know what you're doing then, so they can so it's easier to get feedback, it's easier to communicate. So I reckon it's a two-step. It shows that you've got output, it allows other people to use it, but also it allows you to get feedback for the other ongoing experiments that you're working in. And significantly, yeah, it's just shown that part of what I've been working on during my postdoc as well.
And just before we finish, is there anything that we haven't discussed but you would like to mention?
I think we've covered a lot of it. One of the things is that that doesn't come out in the protocol paper is the troubleshooting that went into it, and the amount of different nuclear extraction protocols that we use. We tried everything. We tried tissue ruptors. We tried like different companies use different tissue ruptors as well. We tried downsing. We tried sucrose gradients. Like there's a lot that didn't go into this protocol as well. And it's just one of the things is that there's a lot of trial and error that you don't see in these publications, but it's there. And to like what I would have liked to have known when I started this, that, you know, it does pay off to just try all these different things beforehand. Eventually you do find something that works, but it can be quite difficult at the start.
Okay, Rose, I think it's been a very interesting and enlightening conversation. Thank you so much for your time.
Thank you, Gabriel.